our next speaker is Jacob Vogel on a molecular gradient along the longitudinal axis of the human hippocampus informs brain function.
I'm just going to test this real quick. Okay, well, thanks for the introduction. I'm going to be talking to you about the hippocampus and sort of the subarchitecture of the hippocampus. And generally, when we think about the subarchitecture of the hippocampus, we're thinking about hippocampal subfields, but there's also this sort of orthogonal axis of organization to the hippocampus, which occurs systematically along the sort of anterior-posterior extent, or the longitudinal axis. And in simple terms, this is a partition between the anterior and posterior hippocampus, but there's also been a lot of work that suggests that behavioral domain specificity and regional connectivity really vary systematically in a gradient moving across this longitudinal axis. And there's been a lot of really excellent studies that have come out recently that have demonstrated this well, where the authors have taken data, whether it's functional connectivity or meta-analytic coactivation or structural covariance between the hippocampus and the rest of the brain. And when they decompose this data into its sort of the components that explain the most variance, always end up either as gradients or partitions along this longitudinal axis, which really again suggests that the anterior and posterior hippocampus are engaging in different systems and ostensibly in different behaviors. So this has also been studied a lot in the mouse. Of course, due to the anatomy, it's not the anterior-posterior axis, but the dorsal-ventral axis. But there's still a longitudinal axis in rodents. And whether it's behavioral domain specificity or regional connectivity, there's actually quite a lot of homology between the rodent and the human, which is pretty cool. But of course, with the rodent, we're able to delve a little bit deeper, start to understand the different genes and proteins that are contributing to this axis, and start to characterize them and learn, exactly what's going on. Obviously, that's a lot harder in humans. So while there's been really excellent work characterizing this longitudinal axis in this sort of functional axis in humans, there hasn't been a lot of work to understand, what are the genomic underpinnings of this. So that's kind of the purpose of this study. We're endeavoring to start to characterize the genomic anatomy of this longitudinal axis in the human hippocampus and try to understand how it relates back to sort of this behaviors and brain connections. So to do this, we'll be using the Allen Human Brain Atlas. I'm sure many of you in the room are intimately familiar with this data set, but there's 3,700 tissue samples that have been extracted from the brains of 6 human donors, each of which have undergone microarray gene expression. And each of these tissue samples also have stereotaxic coordinates that let us know exactly, or more or less exactly, where the probes were extracted from in the brain. So our first sort of objective here was to actually identify this longitudinal axis in the human hippocampus. So we found a plane that stayed within the center of mass of the hippocampal volume, which we're calling the hippocampal skeleton, essentially. And we had 170 different tissue samples that were extracted from the hippocampus. So for each of these samples, we found sort of the closest skeleton coordinate for these samples. And that way we could effectively transform this from a three-dimensional XYZ coordinate space to a single dimension, which is just location along this anterior-posterior extent. We could then use this machine learning approaches to try to predict where along this anterior-posterior axis the tissue samples were extracted from using just gene expression information. And to do this, we used a sparse principal component regression with multiple tenfold cross-validation. So this is sort of our first result. So we were able to predict fairly well where along the axis these tissue samples were extracted from using just gene expression information. We ended up with a mean absolute error of about 2 millimeters. And this is a sort of a rendering of our results on a hippocampal surface, where you can see that the regions that are in red were predicted to be more anterior along this axis, and the regions in blue were predicted to be more posterior. So we did a sort of leave one subfield out analysis where we trained our model on samples from 5 of the subfields and then used this to predict samples from the 6th subfield. And what that revealed was that this sort of gradient was actually quite consistent across the subfields that we had labeled there. And we did something similar with the leave one donor out analysis and found once again the gradient was fairly consistent across the 6 donors. We were also, of course, interested whether this would generalize outside of this particular data set. So we looked to the BrainSpan data set, which has an additional 4 prenatal human brains with gene expression information. We don't have exact stereotaxic coordinates, but we do have very precise anatomical labeling in this data set, so we could figure out which samples were taken from the posterior or anterior hippocampus. And the feature sets were exactly the same as the Allen Human Brain Atlas, so we can simply apply the weights from our model trained on these adults to this prenatal set. We did that. We found that we could get a pretty good separation between samples from the posterior and anterior hippocampus, which was really nice because not only is it generalizing to another data set, but another sort of position along the trajectory of human development, which is nice. So going back to our original model in the adult data set, so we did a sort of feature explainer pipeline to try to get at what are the really core features that are predicting the location of a sample along this axis, and we were able to identify a small set of just 100 features that got an R-square up to 0.8 in terms of predicting location. And when we delved a little bit deeper into this particular gene set, we did some gene ontology and found the set was enriched mainly for terms related to morphological development, which makes a lot of sense given this question. And we also identified a number of genes in this top set of top 100 that had already been mentioned in previous studies of the rodent longitudinal axis of the hippocampus. So that was pretty cool, gave us some insight that we were kind of on the right track. So next, we were interested to learn, so we've identified this molecular gradient, but we're curious as to how it relates back to the rest of the brain. So we had these other 3,700 tissue samples for the rest of the brain, and we sort of put them in our model and said, based on the gene expression of this tissue sample, if this sample were to be expressed along this longitudinal axis of the hippocampus, where would it be expressed? Would it be more posterior or anterior? And by doing so, we have a single measure which tells us if it's larger, it suggests that the tissue sample is more genomically similar to the anterior hippocampus, and if it's lower value, it's more similar to the posterior hippocampus. We called this the hippocampal axis genomic gradient index of similarity, which is really long, so we shortened it to Haggis. And so when we applied this to all the tissue samples across the brain, we saw this kind of striking pattern where these brainstem and anterior ventral regions were more genomically similar to the anterior hippocampus, and these sort of cerebellar and posterior dorsal regions were more similar to the posterior hippocampus in terms of the gene expression patterns. So we were interested, are we picking up just some sort of anterior-posterior gradient, or is this some sort of hippocampal system that we're picking up? So we try to delve into that a little bit deeper. So sorry, this is a busy slide, but on top you see this is that same Haggis map I just showed you, but it's smoothed a little bit just for visualization purposes. And moving down, we have a relative anterior versus posterior functional connectivity between the anterior and posterior hippocampus, relative difference between structural covariance between anterior and posterior hippocampus. In both of those, the anterior is shown in orange. And then we have a map that's showing differential disease vulnerability to frontotemporal dementia and Alzheimer's disease, which are two neurodegenerative diseases that target the hippocampus but target ostensibly different hippocampal networks. And for all these different sort of brain hippocampus maps, we found a fairly strong correlation between our genomic signature and these different systems, which suggests that at least a portion of the variance of these maps can be explained by basically similarity in gene expression. So we're also interested to see if we could find some features that are similar between these different maps. So actually for each of these maps, we did another model to try to pull out what are the important genomic features that predict these maps and to see if there's any overlap. And when we did that, we found a number of genes that seem to participate in all of these different systems. So each one of these genes is one of the top 100 features for the hippocampus model, and it also ended up being a top genomic feature for one or more of the different maps, whether that's functional connectivity, structural covariance, or disease vulnerability. And we were excited to find that a number of these features actually had been previously identified to be related to functional connectivity in humans. So finally, I mentioned that there's this sense of behavioral domain specificity that varies along the longitudinal axis, and we wanted to sort of get into that as well. So here we turn to Neurosynth, their LDA meta-analytic topic covariance map pairs. Basically, there are these meta-analytic covariance maps that are associated with a list of related terms that give you a sense of the particular behavior associated with that map. And we binarized each map and calculated the mean Haggis of all the samples falling within that map. of course, as long as there was at least 500 samples in there. So we could get a sense of, if regions participate in a certain behavioral network, are those regions more genomically similar to anterior or posterior hippocampus? And when we did that, this is kind of just a big summary of the results in one figure, but we found that the more anterior ventral regions, which participated in things like social cognition, conditioning, and memory, and so forth, those regions were also more genomically similar to the anterior hippocampus, while these posterior dorsal regions related to spatial representation and working memory simulation and so forth were more genomically similar to the posterior hippocampus. And this lines up very well with previous literature. So all in all, we were able to identify this genomic gradient that varied systematically along the longitudinal axis of the hippocampus, and this single genomic gradient then was sort of defined these two distinct sort of hippocampal cortical systems, this sort of ventral system and this dorsal system, which we found very interesting. So with that, I'd like to thank you guys for your attention and thank my collaborators and co-authors and our funding bodies, and of course all of these organizations that have put all this data publicly online, without which we just wouldn't have been able to do any aspect of this study. And if you'd like more information, I've got a poster today at 12:45. That's W325. You can check out the preprint online, and all the code used to do all these analyses is also online at GitHub. Thank you.